Hi everyone! Today we'll be starting a series all about biochemistry for the MCAT. This series will roughly follow the Kaplan book since I happen to use them when I studied for the MCAT, and today we'll be going over chapter 1, which is all about amino acids, peptides, and proteins. The reason why amino acids are called amino acids in the first place is because they contain this amine, as well as this carboxylic acid group. And these are functional groups that you should be familiar with from organic chemistry, and in this series I'll assume that you have a basic knowledge of organic chemistry. So all the 20 amino acids that the MCAT tends to test on are known as alpha amino acids, which means that their amine is attached to the alpha carbon, which means the carbon that's one bond away from the carboxylic acid group. Now there are beta amino acids, which have amines that are 2 carbons away, or gamma amino acids, which have amines that are 3 carbons away, but these are not tested on as commonly on the MCAT, and you're definitely not expected to memorize them, because only the alpha amino acids are coded for by our genetic code, which means that we have codons that correspond to each of the 20 alpha amino acids. So this alpha carbon is a very important carbon, and you can think of it as the central carbon of your amino acid because it connects the amine, the carboxylic acid, as well as the R group. And this R group is the one that varies. Another important property of this alpha carbon is that it is a chiral center because it is attached to 4 separate groups. It's attached to an amine, our carboxylic acid, our R group, and a hydrogen. So if you remember how to draw a Fisher projection from organic chemistry, if you put the carboxylic acid group on top, the R group on the bottom, you can choose to put the amine on the left, which will give you an L amino acid, or put the amine on the right, which will give you a D amino acid. And all of the amino acids in our body are found predominantly in the L form. So if you remember how to translate a Fisher projection into a version you're more familiar with, you take the horizontal lines and turn them into wedges, and the vertical lines and turn them into dashes. So here I've drawn out the L amino acid, and as you can see, or if you remember from organic chemistry, all of these L amino acids will be in your S absolute configuration. There are 2 notable exceptions to this rule, and I'll talk about them in the next slide. Here I've drawn out all 20 of the amino acids that you're expected to know and memorize on the MCAT. I know this can seem really overwhelming at first, but once you learn more about biochemistry, all these amino acids will start to look really familiar to you, but you're going to have to memorize all of them, and you're going to have to memorize their full name, their 3-letter code, as well as their 1-letter code. Because on the MCAT, sometimes they only give you the 1-letter code for an amino acid, and so you have to memorize not only the structure, but all of the different names for it. And there are 5 groups of amino acids. The first are the non-polar, non-aromatic amino acids, and these tend to be the simplest. So the simplest amino acid of them all is glycine, and glycine is the simplest because its R group is simply a hydrogen. So this is the first exception to the S absolute configuration rule because glycine has 2 hydrogens on its alpha carbon, which makes glycine achiral. So alanine is the second simplest amino acid, and alanine has a CH3 group as its R group, and as you go down, they increase in complexity till you reach proline, which is a ring. So glycine and proline are commonly found in the loops or in the turns of proteins, because glycine is very flexible due to it having a hydrogen as an R group, which means it can adopt a wider range of angles than the other amino acids can because it has less steric hindrance. And proline is often found in the loops of a protein because proline has a ring that includes its amine group, so it adopts a different angle than most of the other amino acids. The next group of amino acids are the aromatic amino acids, which include tryptophan, phenylalanine, and tyrosine. So an easy way for me to remember phenylalanine is that it has a phenyl group attached to what is basically alanine. So the aromatic amino acids tend to absorb light at lower wavelengths, and they can handle a charge somewhat better than nonpolar, non-aromatic amino acids. The next group are the amino acids with polar side chains. This includes serine, threonine, asparagine, glutamine, and cysteine. Cysteine is the second exception to the S absolute configuration rule because cysteine has a sulfur, which happens to have higher priority. And so although cysteine will be in the R absolute configuration, cysteine is still an L amino acid. There are no exceptions to the L amino acid rule. The next group are your negatively charged amino acids, and these are aspartic acid and glutamic acid. So aspartic acid and glutamic acid are known as negatively charged because they have this extra carboxylic acid group at the end of their R groups, and this carboxylic acid tends to be deprotonated at physiological pHs. So when you have these amino acids in your body, they tend to have a negative charge on their R group. The next group are your positively charged amino acids, and they tend to be positively charged because they have nitrogens in their R group that tend to pick up a proton and become positively charged. Chapter 1.2 is all about the acid-base properties of amino acids. So as we discussed earlier, all amino acids have this amine as well as this carboxylic acid. And if you remember from organic chemistry or from general chemistry, carboxylic acids tend to lose their protons, and amines tend to gain a proton. And these are properties of their pKa. So the pKa of the carboxylic acid group in an amino acid is around 2, which means that at a pH of about 2, this carboxylic acid group will lose its proton and it will become negatively charged. On the other hand, the amine has a pK of about 9, which means that at a pH of about 9, this amine will grab an extra proton and it will become positively charged. So amino acids are amphoteric because they can act both as an acid and as a base. So here I've drawn the 3 possible forms for your amino acids to exist in your body. On the very left, we have what your amino acid would appear as at very low pHs. So this is if you are in very acidic solution, maybe at pH 1, and the concentration of H+ ions is very high. So because we're at pH 1, we're below both the pKs of the carboxylic acid group and the amine, which means that the amine will grab a proton and become positively charged, and the carboxylic acid group will be protonated as well. So this amino acid will have a net charge of +1. So if we then increase in pH past pK1, which is the pK of the carboxylic acid group, the carboxylic acid will lose its proton while the amine will stay protonated, which means that this amino acid will have a net charge of 0, making it neutral. However, it has a positive end and a negative end. So this neutral form of an amino acid is known as the zwitterion, or the zwitterionic form. If we then keep increasing our pH past pK2, which is the pKa of the amine, our amine now deprotonates and our carboxylic acid group stays deprotonated, which gives our amino acid a net charge of -1. So an important equation in this section is how to calculate the isoelectric point. The isoelectric point is simply the pH that this zwitterion has the highest concentration at. So here I've drawn a titration curve for a classic amino acid. If you start at very low pH and you slowly add hydroxide, you'll reach your pK1, which is the pK at which the carboxylic acid tends to deprotonate. And then you'll reach your isoelectric point, and then you'll reach pK2, which is the pK of your amine. So what's really convenient about the PI, or the isoelectric point, is that it lies halfway between pK1 and pK2. So this makes sense because the concentration of the zwitterion should be highest when the pH is between pK1 and pK2. So here I have a question. What is physiological pH? Physiological pH is usually around 7.4, which means that at a pH of 7.4, which is between 2 and 9, the zwitterionic form is most common. So in your body, your amino acids exist predominantly in this zwitterionic form. Now, calculating the PI for your positively charged and negatively charged amino acids is slightly different because your side chains are now charged as well. So if you start at very low pHs for positively charged amino acids, this R group will have a positive charge on it. And The reason why these amino acids are known as positively charged is because the pK of their R group lies above the pK of even the amine. So as you increase the basicity of the solution, this R group retains its positive charge for the longest. So when we start at our most acidic side, this amino acid will have a charge of +2, and once we increase the pH past the pK1, which is the pK of the carboxylic acid, this carboxylic acid will deprotonate, giving it a net charge of +1. Once we increase the pH past the pK of the amine, now you have reached your zwitterionic form. Now this is different from your neutral amino acids, because your zwitterionic form is no longer when your amine is protonated and your carboxylic acid is deprotonated. It's when your positively charged side chain is protonated and your carboxylic acid is deprotonated. So the equation to calculating the isoelectric point is slightly different because your zwitterion, which is when your amino acid is neutral, lies halfway between pK2, which is the pK of the amine, and pKa, which is the pKa of your side chain. The negatively charged amino acids are negatively charged because the pKa's of their R groups is especially low, which means that at very low pHs, the R group will become deprotonated and possess a negative charge. So when we start at very, very acidic pH, our amine will be protonated, our carboxylic acid will be protonated, and the R group will be protonated. And the first pK we'll reach is the pK of the R group, which means the R group will now have a negative charge on it. So your amine will still be protonated and your R group will have a negative charge, giving this a neutral charge. And so now this is your zwitterionic form. So calculating the PI for your negatively charged amino acids is also slightly different because this is the average between your pKa of your R group and pKa1, which is the pK of your carboxylic acid. Chapter 1.3 is all about the peptide bond. So we'll start with some definitions. A peptide is any chain of amino acids. A dipeptide has 2 amino acids. A tripeptide has 3 amino acids. An oligopeptide has 3 to 20, and a polypeptide has more than 20. One thing that I forgot to mention earlier is that amino acids are also known as residues. They can be called amino acids, residues, or amino acid residues, and all three of these terms mean the exact same thing. So to create any sort of peptide, you have to take 2 amino acid residues, and they undergo this process called peptide bond formation. You don't have to know what exactly happens during this process, just that afterwards, you have this bond called a peptide bond, and it is formed between the amine of one amino acid residue and the carboxylic acid of the other. This process also creates a water molecule, and the reverse of this to break down your peptide is hydrolysis. So hydrolysis will take up a water molecule and peptide bond formation will make a water molecule. So when this peptide bond is formed, one of your R groups will lie on the bottom and the other will lie on the top. And when you extend this chain to make oligopeptides or polypeptides, this will keep alternating. One R group on the bottom and one on the top, and one on the bottom and one on the top. So the reason why this is significant is because on the bottom, I've drawn 2 resonance structures of this dipeptide. So the lone pair of this nitrogen can move over here, and the electrons here can kick up into the oxygen, forming this resonance form. And this resonance form is important because it shows that this peptide bond has double bond character. And as you can remember from organic chemistry, double bonds cannot freely rotate. So this peptide bond cannot rotate, and so this R group will stay on the bottom, and this R group will stay on the top. Chapter 1.4 is all about primary and secondary protein structure. So proteins consist of hundreds or thousands of amino acids that all come together and fold in a very complex way in order to do one function, the function that the protein does. So these interactions between different residues are very complex, and so in biochemistry we separate them into 4 different types of interactions. The first is primary structure. Primary structure is very simple. It's just the linear arrangement of amino acids. So if I have a peptide made of 4 residues here, alanine, isoleucine, leucine, and tryptophan, the primary structure is just the order that they're connected in. And on the MCAT, you can expect to see primary structure in the full name form, the 3-letter form, or the 1-letter form. Secondary structure is one level above primary structure. It's the folding patterns between amino acid residues that are neighbors, or they're very close to each other. And the 2 common forms of secondary structure that are tested on the MCAT are alpha helices, which means the residues form hydrogen bonds to form this kind of helical shape, and beta pleated sheets, which come together sideways, kind of like this, to form a planar structure. And so these are 2 ways that you can see alpha helices and beta pleated sheets depicted. They can be depicted like this or like this. Both of these are pretty common. Chapter 1.5 is about the tertiary and quaternary structure of proteins. So it starts off by describing the 2 different categories of proteins. One are your fibrous proteins, which tend to form sheets or strands. Some examples of fibrous proteins are collagen and myosin. So fibrous proteins tend to exist in muscle tissue or in other structural kinds of tissue, and they tend to do movement-related functions. Globular proteins are kind of circular in shape. Examples of these are hemoglobin and myoglobin. So hemoglobin and myoglobin exist in your red blood cells, and they don't really do structural functions. Instead, they can hold on to oxygen molecules. And this is included in this section because proteins are differentiated into fibrous and globular based off of their tertiary structure, or how they fold, how the entire protein folds. So tertiary structure is the 3D structure formed by amino acid residues that are not directly next to each other. One example of this are disulfide bonds. So we discussed before a cysteine residue, which here's the alpha carbon, here's the amine, and here's the carboxylic acid, and it has a CH2 group and then a sulfur. So if you have 2 cysteine residues in your protein, here I've drawn an alpha helix to show that these 2 cysteine residues are not next to each other. They can be very far away in primary structure. And so this can undergo disulfide bond formation, which leaves a covalent bond between these 2 cysteine residues. So now, although these 2 cysteine residues are not next to each other, they're connected, which locks the protein into this shape. Another example of tertiary structure is hydrophobic versus hydrophilic residues. So proteins exist in your body typically in water or in aqueous solution. And so your polar and charged amino acids tend to fold on the outside, and your non-polar amino acids tend to fold on the inside. Now, I have a question here, which is what would happen to tertiary structure if you put the protein in a non-polar solvent? So if you put the protein in a non-polar solvent, your non-polar amino acids will flip toward the outside, and your polar and charged amino acids will flip toward the inside, which will disrupt the tertiary structure. Quaternary structure is when multiple proteins fit together to do one function together. So when multiple proteins fit together, each of these smaller proteins are known as a subunit. So for example, the very last step of electron transport involves a protein that has many different subunits, and I've shown them here in different colors, and they come together to do one function, which in this case is allow H+ ions back into the mitochondria. Other examples of quaternary structure are when proteins are attached to things that are not proteins. So when this happens, these are called prosthetic groups, and some examples are glycoproteins, which are when a sugar is attached to a protein, lipoprotein, which is when a lipid is attached, nucleoproteins, which are when a nucleic acid is attached, and metals can also be attached. So hemoglobin contains iron, and that's why it's called hemoglobin, because another word for iron is heme. Chapter 1.6 talks about denaturation of a protein, which is when a protein breaks down. So some factors that can cause this are heat. So for example, when you cook an egg, you can see the egg white turn from a clear color to an opaque color. And this is because the proteins in your egg whites are denaturing. Another example is when you use harsh solvents such as urea or detergents. And detergents disrupt hydrogen bonding, so this disrupts multiple levels of protein structure. And urea is just a very harsh agent that can disrupt bonding also. And yeah, so in these cases, the tertiary, quaternary, and secondary structure can be disrupted. It's very hard to disrupt primary structure, but that can happen as well. Oh, and if you've made it this far, thank you so much for watching.